Yeah, hi. Something in a way off topic. I'm not a doctor or anything. Uh, I just stumbled across this one. Research reveals surprising findings on how salt affects blood flow in the brain. A first of its kind study. Somehow that's surprising to me. Never ever anyone thought about that. Led by researchers at Georgia State reveals surprising new information about the relationship between neuron activity and blood flow deep in the brain, as well as how the brain is affected by salt consumption. When neurons are activated, this typically produces a rapid increase of blood flow to the area. The relationship is known as neurovascular coupling or functional hyperemia, and it occurs via dilatation of blood vessels in the brain called arterioles. Dilatation is, as far as I know, like shrinking or growing. Functional magnetic resonance imaging fMRI is based on the concept of neurovascular coupling. Experts look for areas of weak blood flow to diagnose brain disorders. However, previous studies of neurovascular coupling have been limited to the superficial areas of the brain, such as the cerebral cortex. Okay. And scientists have mostly examined how blood flow changes in response to sensory stimuli coming from the environment, such as visual or auditory stimuli. Yeah, I bet Slayer raises some people's blood pressure. Listening to Slayer. Little is known about whether the same principles apply to deeper brain regions attuned to stimuli produced by the body itself, known as interoceptive signals. To study this relationship in deep brain regions, an interdisciplinary team of scientists led by Dr. Xavier Stern, professor of neuroscience at Georgia State and director of the University Center of Neuroinflammation and Cardiometabolic Diseases, developed a novel approach that combines surgical techniques and a state-of-the-art neuroimaging. The team focused on the hypothalamus, a deep brain region involved in critical body functions including drinking, eating, body temperature regulation, and reproduction. The study, published in the Journal of Cell Reports, examined how blood flow to the hypothalamus changed in response to salt intake. We choose salt because the body needs to control sodium levels very precisely. We even have specific cells that detect how much salt is in your blood. When you ingest salty food, the brain senses it and activates a series of compensatory mechanisms to bring sodium levels back down. The body does this in part by activating neurons that trigger the release of vasopressin, an antidiuretic hormone that plays a key role in maintaining the proper concentration of salt. In contrast to previous studies that have observed a positive link between neuron activity and increased blood flow, the researchers found a decrease in blood flow as the neurons became activated in the hypothalamus. The findings took us by surprise because we saw vasoconstriction, which is the opposite of what most people described in the cortex in response to a sensory stimulus, said Stern. Reduced blood flow is normally observed in the cortex in the case of diseases like Alzheimer or after a stroke, ischemia. I have no idea how to spell that word. The team doubled the phenomena inverse neurovascular coupling, or a decrease in blood flow that produces hypoxia. They also observed other differences in the cortex. Vascular responses to stimuli are very localized and the dilatation occurs rapidly. In the hypothalamus, the response was diffuse and took place slowly over a long period of time, whatever that means. When we eat a lot of salt, our sodium levels stay elevated for a long time, said Stern. We believe the hypoxia is a mechanism that strengthens the neurons' ability to respond to the sustained salt stimulation, allowing them to remain active for a prolonged period. The findings raise interesting questions about how hypertension may affect the brain. Between 50 and 60% of hypertension is believed to be salt dependent, triggered by excess salt consumption. The research team plans to study this inverse neurovascular coupling mechanism in animal models. Animal models? Yeah, let's build a pig. To determine whether it contributes to the pathology of salt dependent hypertension. In addition, they hope to use their approach to study other brain regions and diseases, including depression, obesity, and neurodegenerative conditions. If you chronically ingest a lot of salt, you'll have hyperactivation of vasopressin neurons. This mechanism can then induce excessive hypoxia, which could lead to tissue damage in the brain. If we better can understand this process, we can devise novel targets to stop this hypoxia-dependent activation and perhaps improve the outcomes of people with salt-dependent high blood pressure. This was really interesting because I was thinking about making a video for a quite long time. I still didn't do it. And that's not it, still. But I was talking with my son about, like, he uses sometimes really much salt, and I somehow tried to explain him some things, but he's a kid. Now I explain to you what I meant. Fastener facts and tool topics. Impacts of salty air on metal building systems and how to reduce them. During a construction project, a wise builder knows that we don't only look at the building materials we are using. I haven't read that article yet. But also the environment of where they will be used. Our materials will react and be altered by fluctuations in temperature, stability, moisture, and other outside factors. One interaction in particular can be the bane of any coastal builder, and that's the interaction of salt and metals. Salty air and moisture can cause what? Salty air and moisture cause metal to corrode and slowly deteriorate. As a result, metal fasteners on buildings in coastal areas can lose their structural integrity from exposure to ocean air. The combination of oxygen, salt, and sodium chloride eats away at the metal, as airborne bacteria from the ocean also consume the iron and cause it to rust. Let's hop over to the next thing, which is G protein coupled receptor. Because I was thinking, like, your thoughts are just electromagnetic impulses interpreted by your brain, which is based on memories, on the past. So let's say this is where the information comes in, and this is where the information goes then to the brain stuff, to the internal web where all the memories live, or something. If they're in between, or if one part of those is not correctly, how to put it, like in functioning order, that there's no way for the electric signal. Maybe I have to draw it bigger. I'm really bad at drawing. Let's imagine those would like that, look like that, and That's the signal, which goes then to the brain. Now, I guess this surface has to fulfill certain properties in order that it can transmit the signal. This surface also needs some kind of surface features which can receive a signal. So probably, since we are talking about electromagnetics, there has to be metal inside those. Metal. Iron, probably, because it's really abundant. Your blood is red because there's so much iron in it. So electromagnetics, iron. And basically, if you talk about Bertrand currents, they are going, I can't really draw that. Like that. Let's try again. Yeah, it's a helix, This is what I'm trying to say. G-protein coupled receptor. G-protein coupled receptors, also known as 7-pass transmembrane domain receptors, 7TM receptors, heptahelical receptors, serpentine receptors, and G-protein linked receptors, GPLR. From a large group of evolutionarily related proteins that are cell surface receptors that detect molecules outside the cell and activate cellular responses. So, spheres and spheres. Coupling with G proteins, they are called 7 transmembrane receptors because they pass through the cell membrane 7 times. Some may claim that 7 is a holy number. Ligands can bind either to the extracellular N-terminus end loops, e.g. glutamate receptors, or to the binding site within transmembrane helices rhodopsin-like family. Rhodopsin-like family. Yeah, must be difficult to listen. They are all activated by agonists. Really? Agonists? That's funny. All through the spontaneous auto-activation of an empty receptor can also be observed. The human beta-2 androgenic receptor in complex with a partial inverse agonist carazolol. So, yeah, I don't have a script, but obviously you can see that they are like spiraling. And this is just, I don't know how exact or how, like this picture, how close it comes to the real reality or how to put it. But anyway, obviously they are spiraled. So, which also is an indicator for electromagnetic activity because, like what I tried to explain before about Bertrand currents, they are also spiraling. So the thing is, if there is imagine this would be a building and this would be a building and there is salt in the air everywhere. Oh damn, wrong button. No, that's one part. I haven't. This is just like, somewhat simplified. S is for salt. And we just read about salt it creates corrosion. So maybe the things just corrode through too much salt. Or there's probably even some accretion on those. Like some other stuff stuck here on the surface. And the signal, which is coming from here and should transfer to the other side in order to get further into our memory and brain that we can make a conscious decision about whatever, or subconscious. But anyway, the transmission could be distorted because those surfaces are corroded. They don't really work properly anymore because there was too much salt. As much as your car standing on some coastal area just gets corroded because of the salt in the air. If you consume too much salt, those transmitters, they corrode, so the thought flow gets disrupted, or the information flow, because of the same reasons your car rusts, or the bridge rusts, which is on near coastal waters. Yeah, I leave it here. Don't use too much salt. That's all. Thanks. Bye.